All right, so I'm recording now. I've taken attendance for those in person, and then looks like most people are all here one way or the other. I wanted to make a couple of announcements with regard to the outline that needs to get done for your research project. The deadline was this Friday. I'm going to push that to Monday. Because of my being ill, I was not able to get to everything as quickly as I wanted to. So I will push that till Monday. And then group one will be in lab this week. So group one, I will be talking with you in person during lab about your research ideas. So before you leave lab, make sure that we have touched base on that and you're feeling comfortable with what you have to do for your outline. Okay, so group one. And then I will continue to answer emails. So, there's a handful, a couple anyways, that I have to respond to and give some feedback. But let's push that to Monday for that deadline. And then I was looking the other day anyways at the forecast, and I know how things quickly they can change with the weather up here, but the other deadline coming up soon is the field guide. It's due not this Friday, but next Friday. I can push that into the following week if you need extra time on that. You have a deadline, right? Is it that 16th for that deadline, or is it?
It's actually the 19th.
19th, okay. So if I push it into like, I could, it's probably better if I do a Dropbox. I'll do a Dropbox for the outline. It should just be, and it's an outline. So you don't have to go whole hog on this part of it. It's more that I want to see that you have a clear question of what you're going to do, and then literally just kind of bulleted points, outline format. This is how I think I'm going to do it. And it is a graded assignment, but it's more of, just do those few things and you're good to go. All right. And then please remember that with your research project, it's a learning experience. I know that a lot of you don't have a lot of micro lab experience, so I want this to be a learning experience. I'm going to be there every lab session. I'm available for one-on-one talks about it. So I really want you to get as much as you can from the experience. And so it's not like I'm sending you off to just do it all in isolation. So please make sure that you understand that. And it's really fun if you let it be fun. It's a good experience. So, and then the field guide, I think I can push that deadline into the following week. So instead of the 16th, if you're still scrambling to get stuff done for that, we can push it to probably that following Friday again. So that would be October 23rd at the latest, because usually snow is not soon after that. And that will give me the weekend to get out there and go tromping around and looking at your sites. It'll take me more than just the weekend. I'll have to tackle it in several sessions, but I like to go out and check out all the sites. So, okay, anything else we need to address? Lab on Friday is group one, and we're going to combine food, and we have to check out our Winogradsky column, see how that's percolating along, and then we'll have time also to talk about the research project and get you going on that. Okay. I'm going to share my screen. So it's been since last week since we've thought about this. Are you seeing one big screen? No? Okay. Let me go back and do it this way. Now is it one big screen? Okay, thank you. All right, so we left off around here looking at carbon cycling, and it's really, really important whether you're thinking about the idea of is oxygen present or is oxygen absent, and then what can happen under those different conditions, different habitats. So I really do want you to get very familiar with this for the next exam. I'll be asking you specific questions on this. So as you're studying, I think it would be a good idea to, of course, work through it, looking at your notes, but then be able to do it without looking at your notes and see if you can sketch this out and get a feel and an understanding for what's happening versus, both with oxic versus anoxic conditions. So we looked at this, and we've covered a lot of this actually here and there through the class already with the when we looked at the physiology section in particular. And so actually a lot of this you've already seen during the physiology stuff, but the new stuff is probably over on to the left here, where we're looking at acetogenesis. We'll talk about that in a minute. Methanogens and methanotrophs. And so the methanotrophs you see here, they're kind of skirting right on the oxic anoxic line. There are methanotrophs that are both aerobic, and then there's actually some species that are anaerobic. So they're trying to show that with both of these lines, and the way it's situated here. Yeah, can you hold the thought? I'm going to show a slide, and hopefully that will answer your question on that. We're going to get into that next. So let's see. We've already talked about the anoxic phototrophic bacteria, those purple and green bacteria that still do their version of photosynthesis, although they only have one photosystem. And they don't use water to extract electrons from water, so that's why they don't create any oxygen. But they're fully capable of fixing carbon in their own special way. And then anaerobic respiration and fermentation. So remember, when you're denied oxygen, microbes certainly can do fermentation. That's probably what you're most familiar with. But we also looked in the physiology section that they can do anaerobic respiration with an electron transfer chain. It's just that they don't have oxygen sitting at the end like we do in our mitochondria. They use something else as a terminal or final electron acceptor. And then I think we left off on this slide looking at methanogenesis. And so far, all organisms that do methanogenesis are anaerobic. They have not found anything that can do it aerobically. And in fact, if you try to work with these microbes in normal lab conditions, they die because they just do not handle the oxygen. They're so well adapted to anoxic conditions. So what are they doing? It's just basically a short electron transport chain that they're doing. And their goal, again, is not to make methane. It's just they're adapted to their particular habitat where, again, hydrogen, remember, hydrogen is just so good, chemically speaking, to donate off electrons. So if they have a lot of hydrogen gas, they'll certainly grab that. They like that. And then you have the bicarbonate ion. And so that's one example. And it gets you enough free energy so that you can make ATP. The magic number for ATP production is if you can get more negative than negative 32. So if you can hit negative 32 kilojoules per mole, negative 33, on up to negative 135 in this case, that's great because you can make good ATP production at that negative number for free energy. But besides carbon dioxide or bicarbonate, you can, when you dissolve CO2 in water, it's bicarbonate. But anyways, you can use other substrates too. It depends on the species in order to react it with hydrogen in order to make methane. So now let's look at the kind of the opposite side. So usually where you find methanogens, you're going to find methanotrophs. Troph is to use. And so methanotrophs are capable of grabbing the methane that methanogens make as waste. And they say, that's not waste for me. I want to use it in order to get my free energy needs met so I can make ATP. So they grab that methane and strip electrons off of it. So here's just an example. And again, I'm not asking you to memorize the equation. It's just to get you thinking about this idea of how they can use methane. And basically, again, they're stripping electrons off of it. So if you can think about that, and then, it's just an electron transport chain process again. And again, the goal is to get that proton gradient so they can make ATP. So methanotrophs can be, depending on the species, they can be anoxic or oxic. They have a little bit more flexibility that way compared to methanogens, but they tend to hang out in the same similar habitats. And remember, just a few millimeters distance for a microbe can make all the world of difference for whether or not they have oxygen available or any other substrates for that matter. So remember, just a few millimeters for them can be a very long distance. So let's look at an electron transport chain. So again, don't get caught up in the details of this. This is just showing how methanotrophs do it in the, the electron transport chain process. So you have methane here on the lower left, and you see that equation working out just like I drew in the previous slide. And I put this here, if you're a visual learner, to get you thinking about the idea that it's an electron transport chain process. That's why they're doing it. It's to strip electrons off of methane. And as they're doing this electron transport chain process, again, it's the same story of there's a proton gradient that gets established during that electron transport chain. And that's great because once you have a proton gradient, you can use that to make ATP. And then you see that they can grab some of these intermediate compounds that are being made through the breakdown of methane and generate their biomass, their carbon-based molecules off of that if they need to build their cells or go through reproduction. Okay, so it's still this idea of the methanotrophs are using methane as an electron source to kick off this electron transport chain. And then just to another visual, get you thinking about this idea of they can coexist. It makes sense if you think about it, that they coexist because the waste by methanogens is the substrate for methanotrophs. And so it's a nice partnership, so to speak, in the sense that they can make use of that waste product methane. And again, it just depends on the aerobic versus the anaerobic, how they run it. But again, it's the same idea. It's an electron transport chain process that they're doing, no matter how they do, which chemical reaction you're thinking of. It's an electron transport chain process. So methanotrophs are good to have around, especially since we know that methane levels are increasing. I wonder if anyone is looking at spiking in methanotrophs into certain habitats where they know the levels are significantly increasing. So cattle are a major source of methane. And I would think someone might have asked that question by now. Could we tweak that? However, I think that'd be a dicey situation with the cattle, because if you start to imbalance their microbiome, it can lead to a lot of issues with the cattle. We'll actually talk about that when we get into microbe-animal interactions. We actually look at the rumen, which is a special part of the intestinal tract of ruminants, including cows. And we'll learn that if you dramatically shift the balance of the microbiome, it can cause death to the cow. So, but could you tweak it somehow and not, be so drastic? Maybe. It might be worth looking into when you think about all the cattle that are roaming the earth right now. That's a lot of methane. Okay, acetogenesis is another thing that was on that flow chart of what's going on, with carbon cycling, whether it be oxic or anoxic. So with acetogenesis, we're back into the anoxic area. And they're making acetate. So Genesis is to create, so they're creating acetate. And again, that's not their goal. It's not like they're like, I want acetate. That's not what they're doing. And so again, hydrogen gas is a favorite thing for them to grab and react the CO2 with. And so CO2 is going to get reduced by hydrogen gas, but they can use other things too. It's not just CO2. And so they're a little bit more broad in their flexibility on how they can run this reaction in order to make acetate. Here's an example. If they do use CO2 and hydrogen gas, so that CH3COOH, that's acetate. But they can use sugars and alcohols in place of CO2. It's good. It has a pretty decent free energy. It's not quite as good as methanogenesis, but it can give you enough free energy to make some decent ATP. And so you tend to see acetogens and methanogens hanging out in the same habitat, including even in cow rumens. And so they're kind of vying for the same substrates. And so there's definitely some level of competition between these two organisms because they're both going after the same substrates. If you look at the free energy, methanogens win. They get better free energy in their reactions, which means then they're more successful. And if they're more successful, they're probably increasing their population a little bit quicker than acetogens. And it's like, okay, so you would think, if you plop methanogens and acetogens together, methanogens should eventually win. And they've actually done these experiments in the lab, and they don't seem like the methanogens don't overwhelm the acetogens, especially if you create a more natural setting and not an artificial, totally artificial system. And so what they realize is that you have essentially, they fall into kind of two categories. The methanogen is efficient. They get good free energy, so they're efficient, but they're more of a specialist. But they're efficient. So they're efficient specialist. And then the acetogens, they don't get quite as much free energy, but they're more flexible. And so they have a little bit more options on what substrates they can pick. And so combining these two ideas, when you mix these two groups together, it works out where they don't tend to overwhelm. The methanogens won't outcompete. And so the acetogens, yes, they don't get as much free energy compared to methanogens, but they're more flexible. And so they can use substrates that methanogens can't. So that's definitely a win in their column in terms of their ability to stay existing in these habitats where the methanogens are found. Acetogens are also a little bit tougher. They can handle lower pH. And so, yes, there's definitely competition, but it doesn't, when you put these two together under realistic conditions, they can coexist. And so there's this nice balance between the two. There's enough flexibility that even though on the face of it, you're saying, wow, they're going for the same substrates. Well, yes and no. There is some nice flexibility here. So you tend to see that in natural settings. In the lab is one thing, but when you get them into more realistic settings, it can end up being much different. Okay. Carbon monoxide, a little bit about that. Lots of it happening in the oceans because of what's going on there with the cyanobacteria and algae. I don't really have time to get too much into this, especially now that I missed Monday with you. So oceans produce, soil tends to be a sink area where it can take CO and fix it through the actions of microbes in the soil. And so the microbes in the soil are, surprisingly, they can take carbon monoxide. It kills us, and yet they can take it and run with it in terms of their physiology and use it as a carbon and energy source. So something that can kill us, and they're happy to work with And it depends on the microbe. Again, I'm not going to ask you to memorize these equations. It's just that you understand that microbes are very flexible here with carbon monoxide. They can do it in the presence of oxygen. They can do it in the absence of oxygen. And so there's different species that can act on carbon monoxide under both presence or absence of oxygen. All right, now there are some very complex molecules. So carbon monoxide is pretty simple. It's a carbon and an oxygen. But then when you start thinking about what microbes can do in terms of recycling really complex molecules, if it weren't for them, our Earth would look so different in terms of just things laying around and not getting broken down, whether it be dead animals or dead plants. And so thinking about this idea of breaking down complex carbohydrates, for example, especially when you get into things like cellulose and chitin, hemicellulose. So cellulose and hemicellulose you find in plants. Chitin is found in insects and fungi. The fungal cell walls are made of chitin and the insect exoskeleton. And so now you need microbes definitely to start acting on this, to start breaking it down and recycling it, the simple molecules through their processes. So again, I'm going to go through these next slides pretty quickly. Remember, these are posted. You don't have to write all this down. Basically, I think, we talked way back when we started this section. There's those tables in the first few PowerPoint slides where, microbes rule when you start looking at this kind of stuff. How do you break down really complex stuff? Fungi are excellent. More and more people are starting to look at fungi as a way to solve a lot of the problems we've created over time with all the pollution, for example. If any of you have heard of Paul Stamets, he actually has a TED Talk. If we can squeeze it in, we'll watch some of that called How Fungi Can Save the World. And so he's a pretty passionate scientist looking at all kinds of fungi and all their applications, including their ability to break down really complex molecules and a lot of things that can kill us, pollutants. Certainly bacteria are no slouch when it comes to this. Streptomyces bacteria in particular, or the group I work with, not only are they excellent antibiotic producers, but they're vital especially in the terrestrial system for breaking down complex molecules. When I was at a scientific conference finishing up my PhD, I was asked to go work with a group for a postdoc out west at the University of Idaho, Moscow, Idaho, looking at how Streptomyces recycle nutrients in the environment. But I wanted to shift my focus more fully into biocontrol. I was very interested in using Streptomyces for biocontrol, so I went that route instead at the University of Minnesota. But Streptomyces are, most antibiotics you've taken to date, you can thank the Streptomyces for that. They are very, very versatile. You can do both aerobic and anaerobic degradation. So again, you can see we're kind of breaking it out by presence or absence of oxygen again. And we realize that microbes can, depending on the microbe, they're very good doing it aerobically and anaerobically. Fungi tend to be more aerobic. There are fungi that can ferment, but for really complex, they can do it quicker under aerobic conditions. But bacteria really shine under anaerobic conditions. So cellulose is part of the cell wall of plants, and the way it's designed, it's a very tough molecule, so to speak, when it's fully formed, and that's why it's part of the cell wall of plants. It really helps them protect themselves, gives them structural rigidity, so that makes it complicated to break down, but the microbes are pretty darn good at it. Hemicellulose, again, is part of the cell wall of plants, and again, it's a little bit more complex chemically speaking. It's a branched polymer. But again, the actinomycetes, including the streptomyces, the bacillus species, all really, really good. And then fungi as well are also really good at this, breaking this down. So more and more attention is being focused on these microbes, especially when you think about bioremediation, all the different pollutants that are very difficult to break down. They have such a long half-life. They're finding that fungi and bacteria can do the job. Chitin, part of insect exoskeleton and fungal cell walls. When you look at it, it's actually, chitin is N-acetylglucosamine subunits strung together in a particular way. And again, microbes, they rule. Breaking this down to simple molecules, they rule. And if you think about all the insects that are out there right now, that's a lot of chitin. Okay. Lignin is the other really complicated molecule that when you think about carbon-based. And lignin is part of what we call the secondary cell wall of plants. And it is even more structurally complex than cellulose and hemicellulose. In fact, they didn't even know the full chemical structure of lignin until not that long ago, even though they studied it over and over and over. Oh, I hadn't heard about that one. Did you hear about that in evolution class? Or? Ah, I'll have to look into that. Yeah, I mean, lignin is incredibly complex. And I can't remember if I mentioned this in this class, but is that paper company still cooking along in Escanaba? There's a paper company there, and when you drive by it, it smells really bad, the chemical smell from that company. And yeah, so they used to be called Mead Paper Company. They've changed names a couple times. I've lost track of it. But anytime you go by, down in Chicago too, when I drive by paper making companies, you could smell that nasty chemical smell. And that's because they have to add these nasty chemicals to speed up the lignin degradation to make paper. Because if you want good quality paper, you have to get rid of most of the lignin. Otherwise, it makes it too brittle and it yellows too quickly. So lignin is just really, really tough. And in fact, at least so far, they haven't seen any organism that can attack lignin under anaerobic conditions. So that's an interesting, maybe that'll evolve eventually, but at least so far, no one has yet to find that. So that's kind of an interesting thing. Now, there are some fungi that have been studied pretty heavily for their ability to degrade lignin. And we actually give them names. We break them out into different groups. So if you look at brown rot fungi, and that's actually what we call them, they're called brown rot fungi because they're the ones that preferentially attack cellulose and hemicellulose. And then the white rot fungi, they preferentially attack lignin. And so the wood, when it's breaking down, will look different. You'll either see a predominantly whitish look to the wood or a brownish look to the wood, and then you can tell whether or not who's acting on the wood and breaking it down at that time. And so that's been heavily studied as well. And then there are aerobic bacteria that lend a hand with this as well. It's not just the fungi. But fungi are really good at it. Fungi have lots of interesting, bizarre enzymes that are super good at breaking down complex molecules.
But that's in aerobic, it's in aerobic conditions. They can't do it without oxygen.
The lignin?
Yeah.
Right, yeah, it's aerobic. So when I was a PhD student up at Michigan Tech University, part of my work was up in the forestry department. And so I took a lot of forestry classes, and They get way into this kind of stuff, looking at the types of fungi and how they're breaking down the wood products. So here is just the partial lignin, just throwing it up there to impress on you. It's complicated. It's not an easy structure. And that's why you have to really throw some nasty chemicals at it if you're trying to make paper, for example, and want to get rid of the lignin. Go into aquatic systems now, out of the terrestrial environment. Polysaccharides are not quite so complex there because there's just, lack of trees and wood products. And so you don't have the complexity and you don't have the amount compared to a terrestrial environment. But bacteria really shine in these conditions because bacteria are pretty good at sopping up really dilute carbon and getting it efficiently into their cells, and then of course using it to grow and produce more of themselves. And so going into the oceans and aquatic systems, bacteria really dominate when it comes to breaking down the polysaccharides. Okay, what else do we have? Oxygen cycle, and I'm not going to say too much about that. The oxygen and carbon and water cycle, it's hard to separate all these out. I guess one of the big things to remember or to think about is, yeah, definitely we have a lot of photosynthetic organisms. Plants, of course, come to mind, but now that you're in this class, we also know that there's the algae and the cyanobacteria that are producing oxygen. So you certainly have production of oxygen going on quite a bit. And then of course there's use of oxygen by all the aerobic organisms out there. So I guess one of the big things I want you to think about when we talk about oxygen is that profoundly influences how microbes will run their physiology. And, if you go back again to that graphic just a few slides back where we looked at what's happening oxically, what's happening anoxically. And so, and again, if you're smart, if you're going to be a smart microbe, if oxygen is available and you can use it, you've adapted to use it, then use it. It is absolutely the best option because it's a great electron acceptor. As far as we know, it is the best electron acceptor out there on the planet. And so that would be a great option because the payoff is good. You get a lot of ATP if you stick oxygen as the final electron acceptor or the terminal electron acceptor during respiration. We do that in our mitochondria, but remember, microbes don't have mitochondria, but they can pack all this kind of stuff into their cell membrane and go at it that way. Now, if you're a microbe and there's no oxygen around you, well, hopefully you have the flexibility to do the next best thing, which would be anaerobic metabolism. Some people say anaerobic respiration. But remember, anaerobic just means now you're doing an electron transport chain. It's just you do not have oxygen as the terminal electron acceptor. You grab something else from your environment. So maybe it's nitrate. That's a really common, some kind of nitrogen containing compound is really, really common. But so are the other ones that I've listed out here, sulfate, ferric, iron. Ferric is the Fe3 plus form of iron, manganese. All of those work really well. It just depends, again, on the habitat. Where is this microbe living? What is it exposed to in its habitat? And that will determine what it's going to grab and use in place of oxygen. So it's, analogous. You have some different players in the game, but it's still an electron transport chain process that you're looking at. And it's just that you don't have oxygen sitting at the end. And then if you're a microbe and you're like, okay, I don't have oxygen, I can do anaerobic respiration. I'm going to grab nitrate. But let's say you hog all the nitrate and now you're depleted it. Your habitat has nothing that you can use for anaerobic respiration. Well, hopefully then you can switch to fermentation, which still gets you some ATP. It's just not great, but it keeps you alive. Maybe even a little bit of reproduction if you're frugal. And so fermentation still works, and some microbes have to do this. It's like their only option. So it just depends on the microbe. Methanogenesis is another option that is anaerobic. That gets you some pretty decent ATP actually. So we talk a lot about fermentation, but now that you've been introduced to methanogenesis and acetogenesis, those could be options. They're just very specialized because there's not many microbes that can do methanogenesis and acetogenesis. There's just a handful in the grand scheme of things. Way more microbes can do a bunch of different fermentation pathways that we've already kind of hit on a little bit when we looked at the physiology. Okay, so again, this idea of presence or absence of oxygen, because oxygen is just so darn good at being a terminal electron acceptor. You get tons of ATP, well, not tons, you get the most ATP doing it this way than any other strategy. So we've looked at that already. I'm just putting this in here to refresh your memory. You've seen this before, where on the left, you have oxygen as the terminal electron acceptor, and on the right, it's nitrate. So on the left is aerobic respiration, on the right is anaerobic respiration. And you can see there's slightly different players in the game, but it's still an electron transport chain process. And then you've seen this, fermentation ideas again, where it still gets you some ATP, and there's so many different options out there, and probably more we have yet to discover on how they can run these different fermentation pathways. But the goal is, you know, they don't care about alcohol like we do. They excrete it out, it's waste to them. In fact, it kills them eventually if it builds up in their environment. That's why when you brew beer or make wine or whatever, there's only so much alcohol you can get from a living microbe, and then they end up just dying in their own waste. They're trying to engineer yeast to tolerate higher alcohol concentrations, and they have had some success, but sooner or later they die because it's just waste to them. They don't care about the alcohol. It's the ATP they're getting. So here we're looking at turnover rates. If you're interested, I'm not going to really ask about this, but the only point here I might ask about would be CO2. You can see CO2 by the way they've drawn it. It's the smaller pool. So CO2 is more easily impacted by activities as, you know, things can get imbalanced, I guess, a little bit easier. So perturbation is the word they use. That's a good word. Perturbation of cycling is most likely to affect the relatively small pool of CO2 when you could do the comparison here. Okay, so carbon cycling, yes. Nitrogen is the other thing to think about. And microbes are absolutely critical for this. You cannot run this cycle without microbes. Unfortunately, the other downside, well, it's not really a downside. Microbes are great. They can recycle. That's awesome. We as humans have impacted the nitrogen cycle pretty strongly by our activities. And so that's led to some problems and pollution. So let's say a few things about it. So with this is just a schematic trying to put it all into one little picture of what we got going with all the different processes. And so I'm going to zoom in to a couple ideas here of how microbes are cycling nitrogen. So remember, our atmosphere is composed of N2, dinitrogen. So that's an N with a triple bond to another N. Very difficult triple bond to break. Only prokaryotic organisms can do that. They're the only ones that have the enzyme that can convert dinitrogen gas into, typically the first product is ammonia, but then very quickly it gets assimilated into some other nitrogen containing molecule. So where is, and then, okay, so then we're going to see how some microbes take dinitrogen and fix it into more complicated nitrogen forms. Other microbes will do the opposite essentially and create dinitrogen gas. And so let's take a peek at that. So here, I do need you to know this word, denitrification. Please memorize that. Denitrification is what is being shown here to completion. If you take some nitrogen-containing compound, and the idea here is that eventually you get dinitrogen. So if you say, oh, that's denitrification, what you're saying is there's some nitrogen-containing compound that's getting converted ultimately to dinitrogen gas. So here, they're showing kind of the starting point as nitrate in this pink box, nitrate here, and you see nitrate reductase. And so E. coli can do this. This is, again, electron transport chain process. E. coli says, I don't have any oxygen to put on my terminal electron acceptor, so I'm going to grab nitrate. So they run that reaction, and in the process, they'll make nitrite, NO3 or NO2 minus. And then some other microbes say, oh, I can keep going. I have the necessary players in the game to just keep running this electron transport chain and keep my proton pumping going. So I'm going to just keep doing it. So there are some microbes that can take it all the way to dinitrogen. And then there's some microbes that can only take it part way. All right, so it just depends on the microbe, but there are definitely microbes that can go and take these nitrogen containing compounds and drive it all the way to dinitrogen. So then that's good because you have to have this recycling of dinitrogen. And then other microbes take dinitrogen, the very special nitrogen fixers, and then they take that dinitrogen and fix it back into a compound that can be used. So depending on the microbes, some microbes, as we talked about in Plant Phys, they do this in combination with a plant, hang out in the roots of the plant and do it there. Other microbes can be free living and just say, I'm going to fix nitrogen all on my lonesome. I don't need a plant to do this. I'm just going to do it. And so it just depends on the microbe. They can be free living and they can take dinitrogen and fix it into ammonia and other organic compounds then beyond that. And so it's really dinitrogen that microbes can grab and use. But again, it is only prokaryotic. We have never found a fungus that can do this, that can take dinitrogen and fix it. Just haven't found it. Maybe someday that'll evolve. I think that'll happen long after we're all dead, but could happen. We have influenced the nitrogen cycle a lot through agricultural strategies and our nitrogen fertilization applications. That's really skewed how much nitrogen and forms of nitrogen. Gram negatives. I'm trying to think of a gram positive example. I'm coming up short. Let me double check that. I think so far gram negatives, but I could just be not thinking. Let me double check that. And this is very classic, the nitrogen triangle. So I like this for studying. I think this says it very nicely. You have your dinitrogen up in the atmosphere, and the fixation idea is off to the left, where very special microbes, whether they're hanging out with plants and doing it in the roots or just free living, primarily in the soil. A lot of these microbes are in the soil. You can take that dinitrogen and fix it. Again, the first product is usually ammonia or ammonium, but then that usually gets quickly incorporated into some organic molecule. Nitrification can then take ammonia and run it to nitrate, for example, or nitrite. And then denitrification means you're creating nitrogen gas again. So I would like you to know this triangle. So please spend some time studying it and either be able to draw it out in shorthand, or if I give you a partly filled out form, you know, be able to put in the terms appropriately. But I think this is really a nice, succinct thing to think about. It's not overly complicated, and it does a nice job of showing the nitrogen, the flow and recycling of nitrogen. So this is really simple. The next slide is a little bit busier, but again, for study purposes, especially if you're visual, you can see all the different ways that microbes, okay, look at, it's all microbes that you're looking at here that are working on the different nitrogen compounds. So this is saying pretty much the other, what we just looked at, it's just a little bit more filled out. Okay, so in lab on Friday, and then the next group, when you get your turn at looking at the Winogradsky column, I'll mention just a couple more things about recycling of nutrients, because in those Winogradsky columns, we do have cycling going on, probably some nice sulfur cycling going on. So I'll touch on that in the big picture idea. So you'll still get a little bit of microbial biogeochemistry in lab, but this is the end of the content in the lecture portion. Now the next topic we're going to get into is microbe and other organism interactions. So we're going to start with thinking about just general concepts of how they interact, and then start looking at very specific interactions with plants and animals. Okay, so and insects, we'll look a little bit at that too. But I guess we're out of time, so we'll stop there.